certainly what is advertised should match what's in the product. And from the consumer point of view, not only is it not matching, but also they could be potentially purchasing some dangerous ingredients.
Yeah, and that's when the term Psychedelics was coined. So that's synthetic psychedelics.
Welcome to the Pittcon podcast, where we hear from world-leading scientists about their journeys from passionate young academics to professors to Pittcon presenters. Today we speak to Dr. Jonathan Ferguson, scientific business leader at Shimadzu Scientific Instruments, about a growing public health concern that first came to light at Pittcon 2024, synthetic psychedelics. Jonathan shares how an investigation into psilocybin products revealed unexpected and potentially dangerous compounds, leading to collaborative work with Rose City Laboratories and Oregon State University to better understand what's really in these unregulated products. By the end of this episode, you'll understand how analytical chemistry is uncovering hidden risks in emerging psychedelic markets, and why collaboration across science and regulation is essential for protecting public health.
Jonathan, thank you for joining us today on the Pittcon podcast. Before we get started on the topic of today's episode, which is synthetic psychedelics, could you introduce yourself and your role at Shimadzu and what you do there?
Sure. I'm Jonathan Ferguson. I'm the scientific business leader in the new strategy department at Shimadzu Scientific Instruments. I'm based in Columbia, Maryland.
Perfect. And you were here at Pittcon last year.
Correct.
Can you tell us more about the symposium? So it was the Psychedelics 101 symposium. Who was there? What was it like? We were in San Diego then. Tell us about that session.
Sure. So in 2024, Pittcon had their first psychedelics symposium. I was the second speaker at that symposium, and it was designed around quality control of naturally occurring psilocybin-containing compounds for the purposes of clinical trials in depression.
And they're like psychedelic mushrooms?
It could be mushrooms, yes. Yep, mainly mushrooms.
Mm-hmm.
Certainly you could put them in chocolates and gummies and that kind of thing. And there's reasons for doctors to do that. So I was the second speaker at that symposium, the first being Frisch USA. They talked about good milling strategies and the need for good sample preparation. And then myself. Then the third talk was Scottsdale Research Institute. And the fourth was Rose City Laboratories. Now, at that fourth talk, Bjorn Fritzsche, he's the senior R&D scientist at Rose City, he was running some samples and he found some additional peaks that he couldn't quite explain. So natural scientific curiosity, he ran them on his GCMS, that's gas chromatography, mass spectrometry, and he was able to identify cannabinoids and horse tranquilizers, which wasn't supposed to be in the sample.
Yeah, and so these gummies are things that people can buy freely from like convenience stores or even online. So the fact that they have these compounds in is really significant from like a public health perspective.
Correct. So you can find them at gas stations and head shops in Oregon where psilocybin has been legalized at the state level. You just go in there, you buy a package, and what we're discovering is what's on the package is not matching what the product actually contains.
Yeah, and this was the first time this had been discovered at Pittcon last year. Is that right?
We were surprised to see it. It can be a bit of a wild west with a lot of moving targets, but certainly what is advertised should match what's in the product. And from the consumer point of view, not only is it not matching, but also they could be potentially purchasing some dangerous ingredients.
Yeah, and that's when the term Psychedelics was coined. So that's synthetic psychedelics. And that's sort of the area of research that you're going into. And there was a collaboration that was founded. Can you tell me more about that?
Yeah, so after Bjorn Fritzsche discovered these, we wanted to pursue this a little further. So we were able to connect Rose City Laboratories with Richard Van Bremen at the Linus Pauling Institute to do some deeper analysis and confirmation of what was seen in a variety of samples. So it was interesting because this symposium was supposed to be about quality control methodologies in ORLAP accredited labs. And what became of it was this discovery of synthetic psychedelics or syndelics in these products, where consumers are buying these things at head shops and gas stations where what's on the package doesn't match what's in the container, and they're potentially purchasing some dangerous ingredients. So we are working on a manuscript to publish in a peer-reviewed journal, and we'll be presenting some of this data in future conferences. And the data is very interesting, but also a little bit alarming.
Mm-hmm. And you can't tell us too much, but it's looking like this is an emerging public health threat.
I don't think a lot of people really know. There's not a lot of regulation on some of these products. You can actually even attend this conference. Just yesterday, they're talking about how you have some products that are legitimately made, and also a lot of contraband, where what's in them is not really known, and they can look very similar.
Mm-hmm. And also at the show, we've got talks this year on, let's start again. I've gone blank again. It's on, yes, I've remembered. And at Pittcon as well, we've got some talks on edibles, cannabis edibles, and the regulation of them. How significant is Pittcon in facilitating these conversations and bringing these issues to light?
Well, I think it's very important. So you and I just walked here and we saw the exhibition hall, and you look out and it's very impressive with all the marketing and marquees. But Pittcon is really more than just an exhibition hall. It's this hub of collaboration and innovation that really allows researchers and customers and companies to come together and work on, mutual problems and publish research together. And without that forum, these types of relationships wouldn't be able to occur. So I like to thank Pittcon for establishing that environment where we can come together, work together on science that's not only interesting, but meaningful and really has an impact on everyone.
Yeah, definitely. And this is a great example of, this was Pittcon a year ago, but now we're seeing what happened, a collaboration that was fostered a year ago. We can see where it's at a year later. That's an amazing thing to see.
It's always nice to see a year later you further along in a project than where you were a year ago. Yeah. These things do take time, but the wheels are turning. It's very important for us in terms of, we talk about this public health hazard, what are we going to do about it? And what Shimadzu Scientific Instruments can do is just to get the information out. So have the data grounded in good science, present it, publish it, let everyone know so that we can address these problems from a view of a good factual foundation. Yeah, I think that's most important.
Yeah, definitely. And obviously you've got yourselves, you've got Rose City Labs, and then you've got the university as well. So it's a bit cross, you've got an academic institution, research institution, you've got companies. What's that been like with the collaboration?
Yeah, so the way I often will tell this story is, the days of the chemist working alone in the basement, doesn't really happen anymore. Maybe it happens illicitly, but not in the real world. You really need a multitude of researchers to work on these projects because we all bring in different expertise. Without these collaborations, which is the environment which Pittcon provides, we wouldn't be able to move science forward as quickly as we do.
Yeah, and what sort of technological advancements are needed to be able to detect these, worrying substances that are hidden in these products?
So it's the moving target, right? So we talked about these head shops and gas stations selling these gummies and chocolates, and they have who knows what in them. Well, that presents some different challenges. You can look at it different ways. One, you actually have the matrix, so that's the gummy, the chocolate. They're going to have different compounds, which are just naturally supposed to be there, but they can interfere with your analysis. So how do you prepare that sample so that you can target what you want to target? Another problem would be just if you have an adulterant, what is being put in there? Because it's not being done in a regulated way. You don't know what the chemist who made that sample, what they introduced into it. And there can be a variety of variations and substituents on these molecules. And this presents a real challenge that was being talked about at this conference. There's so many variations and you don't know exactly what you're going to see. So famously, this is called the unknown unknowns, where we have to kind of look for it, but we really don't know what to expect. And they can change from sample to sample.
So some big challenges ahead, and another big challenge would be regulation. With the psychedelics market set to grow in the US, what are the major challenges with regulation, and what would you like to see change?
Yeah, I mean, there's a large history there. Just to give you a little bit of history on this, I usually kind of start talking about it for the United States in the 1970s, because you had this hippie culture, which things are kind of increasing. Then psychedelics were listed on the Schedule One as a Schedule One drug, which meant that it had no known medical uses, and that basically stopped all research for 30 years. In the 2000s, things started up again. Some companies started doing a little bit more research, finding out what medicinal uses these things had, and that culminated into, in 2019, Denver, Colorado decriminalized psilocybin. That was quickly followed with Oregon and Colorado State decriminalizing and legalizing, and Oregon setting up a regulatory framework for psilocybin. But it doesn't change the fact that it's still illegal on the federal level. Shimadzu Scientific Instruments, we always do everything completely legal through legal pathways. That's why we worked with Rose City Labs, who is licensed to run these samples, and Richard Van Bremen, who also has a license to do the analytics. So we can't always do everything ourselves. There's a lot you can do with just standards, but having these collaborations, particularly in the psychedelics world, is why it's so important, because you are kind of confined in how you do these in a legal way. In terms of what regulations should exist, I'm not probably the best person to answer that. I can tell you, I will give you my philosophy, and that is people should know what they are consuming, and what is on the package does need to match what is in the sample. And whenever you have a mismatch in any way, whether it's not correct or you can imagine that the dosing could be 10 times as high, or you could have some dangerous compound in there. I mean, in all these scenarios, that's not good. They need to be accurate.
Yep, definitely. And I know you mentioned that you're presenting the results, and they're also being published, but what else is next for this collaboration and for Shimadzu?
Yes, I mean, part of it is going to be driven by the science. As you saw when we started that symposium, we wasn't going to go start looking at gummies and chocolates from head shops. It's just what the science directed us to. So one is what the science reveals. Another is we are always following what customers need. When there is a need there, Shimadzu Scientific Instruments will come in to fulfill it. So it will be driven by regulation that's beyond our control, but we're always trying to meet our customer needs and seeing what's coming over the horizon and preparing for it.
Yeah, and after the results are published and presented, what do you think is next for this collaboration? Do you see this getting a big reaction?
Well, I think so. I think it would be a big reaction because people, I mean, it can be dangerous, and it needs to stop, in my opinion. So I think it'll be picked up, and people are going to realize, oh, wait, we need to address this. How that will be done is kind of beyond, it's going to be outside my hands.
But we're interesting to see how it evolves going forward, because this has been, we're right at the beginning of this field of synthetic psychedelics. So it's going to be really interesting to see how it evolves.
I don't see it stopping personally anytime soon. There's always going to be something else. If you just look at we have a history here with cannabis, and psychedelics is getting a history, and it's always, one more thing. If you look at everything we've looked at in science, and whether it be pesticides or PFAS, it's always something else, and it really never seems to stop. I can't always predict what that something else will be. Sometimes I'm right, I kind of have an idea, and other times like, whoa, that's surprising, we're gonna have to work on that really quickly. But we stick to our fundamentals and keep reviewing the literature, keep establishing these collaborations, so we can kind of predict the future as best as anybody who can predict the future can, and really just stay ahead of things. So that's what we have to do.
And ultimately, it's so important because these are people's lives, like people who are taking these and not knowing what's in them. They could have like, adverse health reactions to some of these substances.
Yeah, certainly. In cases where if if a little bit of fentanyl is thrown into one of the samples, there's cases where people have taken poison pills where there was one pill had too much fentanyl. And fentanyl is not even the worst. If you look at carfentanil is 100 times more potent than fentanyl, which is 100 times more potent than morphine. So if you're good at math, carfentanil is 10,000 times more potent than morphine, and morphine seems to be pretty potent. If that ends up in a sample, that can be very dangerous. So, people need to know what is in these samples and, be careful. They need to be accurate.
And so in this collaboration, what Shimadzu technologies have been used?
Right. So in the research area, LCMS is the liquid chromatography mass spectrometry. I would argue it's still the gold standard. When I was talking about Bjorn Fitzja and his analysis, he did gas chromatography mass spectrometry, also a really good technique that goes hand in hand. Richard Van Bremen has a quadrupole time of flight, a Shimadzu quadrupole time of flight, because it has higher resolution, higher mass accuracy. And that is really useful when you're dealing with the unknown unknowns that I've talked about. Now, when that methodology gets kind of propagated, a lot of times it can be overkill to have that high-end technology, and a lot of people resort to a triple quad mass spectrometer instrument to do the analysis. When I was doing the second talk at the symposium, my focus was actually to provide a methodology that only used LC UV, so liquid chromatography with ultraviolet detection. And that's just simply because, it's cheaper. And if once you refine that methodology and you can get it down just to UV detection, which is not as specific, but once you work everything out, you can run these samples and it's a lot more cost effective. Because it doesn't do a lot of good if only the best scientists of the world with the most expensive equipment can do the analysis. Because ultimately, we'd like to save lives, and if only a couple people, a handful of people in the world can analyze the samples, it's not going to be helpful at, getting the results out. So using methodology that is inexpensive is very important. So Shimadzu Scientific Instruments, we're a leader in liquid chromatography and mass spectrometry. But thinking even beyond that, I was at a talk where they had to come up with a method, and one of the requirements was not to use power. Well, I don't think anyone has developed any LCMS that doesn't use power.
Why was that a requirement?
Well, so let's say you want to test a sample, and in some areas of the world, they just don't have access to reliable electricity or any electricity. And at the moment, I don't know anybody who makes a LCMS that doesn't require electricity. It's kind of fundamental. So in those situations, those customers they have a different set of requirements, just like we have the researchers who need the high-end technology and people in applied market or like at the talk I was giving at the symposium in 2024, I was presenting on methodology that only made use of liquid chromatograph and UV, so no mass spec required, simply because mass spec is more expensive. Now, UV is not as accurate in that you don't have the mass to charge, but once you work out all the retention times and you know what the absorbance profiles look like, or you using, we have a technique called IPDA that helps us clean up the chromatogram, you can get very accurate quantitation results, and it's going to be a lot cheaper. And simply put, that some people in that market, they just don't have the the finances to purchase a mass spectrometer, but they still need accurate quantitation. So LC-UV provides that. Similarly, in some places, they just don't have access to electricity, and those customers have a different set of needs. So we have to be mindful of that they might need to do a sample preparation to determine the quantitation, whatever their analyte might be, but not have access to electricity. It would have to be like antibody-based, something, a color change. Because ultimately what we want to do is keep people safe by providing accurate information. You can also especially see that in the environmental world, right? Obviously, clean water is critical, right?
And a lot of the places that have problems with clean water might not have access to electricity.
Exactly. Yeah. It's something that we have the luxury of not always having to worry about, but some people do, and we need to meet those customers' needs as well.
Yeah. So when did you first start getting interested in science, and how have you gotten to the point where you are today?
Yeah, a lot of people have this moment in their history where like this was the flashbulb moment. It didn't quite work like that for me. I kind of came from the just middle of nowhere in Illinois. I mean, that's not meant to be an insult, but the county where I grew up had one stoplight. Okay, the town where I grew up has 250 to 300 people. It's small, and science provides answers, and not just answers, but answers that if anyone were to reproduce correctly, they all get the same results. And that was really appealing, that you have this strategy of determining how things work. And I guess if I had to summarize it, I love learning how things work, and it can be extremely exciting when you do a discovery and you find something about whether it be a protein or some mechanism, and you now know the answer, and yet no one else in the world does yet, and you contribute to that knowledge, and it's very fundamental, and I found it quite appealing. I also I really like technology. I am like a techno enthusiast. I like working with the big, expensive mass spectrometers. I love tinkering. I came from Professor Neil Kelleher's lab when he was at the University of Illinois in Urbana-Champaign. And I remember walking to his lab, and he has the FTI CRMS, the Fourier transform ion cyclotron resonance mass spectrometer. You can see why we abbreviate it. It's this big 8.5 Tesla magnet. You walk in, and everything, there's like cords everywhere because it's all it's hand built. It's not commercialized yet. It's not in a neat box, and you get to tinker with everything. And so I was allowed to, fix it, which is a blessing and a curse. You have to fix it, and it's work, and it doesn't help you graduate any faster, but the amount you learn is just incredible. And it was very appealing we are literally controlling ions and manipulating them and breaking them apart and using software to put them back together to tell a story. And so he was big in top-down proteomics, which I don't want to spoil what that is. Just go check out his podcast, because I know he was the first podcast you did, and he will explain it far better than I. But just being able to do that is incredible, and I really enjoyed it, and it really kind of fits to my nature.
Yeah, it sounds like it was really hands-on. I can imagine that was really beneficial to be a early student.
Yeah, I like taking things apart. I like knowing how things work. If you understand how things work, you can fix them. If you can fix them, you can get your job done. Nothing's worse than, oh, well, that's not broken. That's not working. That's broken. I guess we're done for the day. Or I could fix it really quickly, and we can continue and get stuff done. And to do that, you really have to know how things work.